When Gregor Mendel established the foundations of genetics, he uncovered a strict biological rule. Inheritance is a 50-50 coin flip. Every time an organism reproduces, its offspring has an equal half chance of inheriting any specific gene from either parent. Because of this 50-50 split, a single new genetic mutation introduced into a massive wild population has to fight a steep uphill battle. Over successive generations, the new trait is diluted and eventually swallowed up by the vast sea of normal DNA. This biological math makes changing a wild population through selective breeding incredibly slow and usually temporary. Nature's strict 50-50 rule acts as a defensive wall to keep species stable, and that stability is the exact reason humanity has historically failed to permanently eradicate fast-breeding threats. For decades, we tried to control rapid breeding pests through massive poison campaigns. But the pests simply adapted, rapidly developing resistance to the harsh chemicals, while the toxic runoff devastated local ecosystems. The human toll of these surviving pests is staggering. Mosquitoes carrying malaria and dengue fever still kill hundreds of thousands of people every single year. In agriculture, herbicide-resistant superweeds like Palmer amaranth choke out entire harvests, causing billions of dollars in economic devastation. Then, researchers like Jennifer Doudna pioneered the use of CRISPR. This molecular tool gave scientists the ability to build theoretical gene drives, mechanisms designed to bypass Mendel's 50-50 math entirely. We are moving from external chemical warfare to internal biological hacks. A CRISPR gene drive converts a single organism into an automated, self-replicating gene-editing machine buried within its own DNA. When an edited organism mates with a wild one, this CRISPR machinery wakes up inside the newly formed embryo. It physically reaches across to the normal DNA strand inherited from the wild parent and slices it in half. The cell attempts to repair the cut strand. To do that, it uses the intact strand as a template, tricking the cell into copying the drive sequence straight across the gap. Because the gene copied itself, the mathematical outcome changes instantly. The offspring's chance of inheriting the edited trait jumps from 50% to nearly 100%. Every generation repeats the process. This automated copy and paste mechanism transforms a localized genetic edit into an unstoppable chain reaction that can consume an entire wild species. Scientists are testing two primary strategies. The first is population replacement. Researchers slip a malaria-blocking gene into the drive sequence of a mosquito. The mosquito species survives and functions completely normally in the ecosystem, but the disease they carry is eradicated. But the alternative strategy is known as population suppression. By forcing a sterility trait or an all-male bias into the gene drive, the target population loses its ability to reproduce. Within a few generations, the species crashes to zero. Humanity now possesses a toolkit to either permanently domesticate a wild pest or intentionally drive it to total extinction. The real test of this technology happens outside the sterile confines of a laboratory. Consider an invasive, non-native mouse destroying an isolated island ecosystem. Deploying a suppression gene drive there could save the island's native birds and plants. But if a single edited mouse sneaks onto a cargo boat and escapes to a mainland ecosystem where its species is natively vital to the food chain, the drive will continue replicating. Because biology ignores borders, a localized experiment can become an uncontained global event. To prevent a runaway scenario, scientists are trying to build the brakes. They are developing self-eliminating drives that are programmed to burn out after a few generations. They are even designing neutralizing elements, literal anti-gene drives deployed into the wild to chase down and overwrite rogue edits. Just because humanity has compiled the code to rewrite the natural world, who actually holds the authority to press enter? Gene drives are simultaneously our most promising tool to save millions of human lives, and our most dangerous capability to permanently alter the Earth's biosphere. If you want to keep exploring the incredible and terrifying ways technology is rewriting our world, subscribe to the channel.